Just before we start going through some of the answers and how to get to the answers using a bit of technique, I would like to give you a couple of tips for any of the three papers. So AQA Biology is paper 1, paper 2 and paper 3. Now it's worthwhile just taking a deep breath obviously before you start any paper and try to relax as much as you can. If you just have confidence in the fact that you've gone through the specification, now they're only going to ask you questions that are based on the spec. They are not allowed to ask you anything else. You might get some AO2 application questions, but basically the answers will be, as long as you use the information that you're given in that particular question that we'll look at during this video, it's going to really tease out of you what you know in relation to the specification. Okay, so as long as you're happy with the spec and you've been through each key point on the specification for each topic and you're willing to use the information in the question, you should be absolutely fine. Now, I would recommend that you skim through the paper first. So when you start the exam, have a quick look through each question just to see what topic is on each particular question. You could then even highlight which topic is on which question. For example, you might have a topic that you're particularly strong with that you might want to start on. I would also recommend having a quick look at the end of the paper for paper 1, because there's normally 1 or 2 5 to 6 markers at the back of the paper. So what you don't want to do is get through the paper but rush the last 1 or 2 questions if there's questions that you can answer fairly easily if it's recall. Right, probably the most important tip I'd give you is to make sure that you really focus on the information that you provided in the question. So you need to understand the context of each question. Now you might read through the text and circle some of the key points. Now you could even simplify that by making little notes of your own next to the circled bit of text. Now in addition to circling key points in the text that are going to tell you what the experiment is about, it's really important to use headers from tables, use the labels of graphs, and also make sure you look at the key and never neglect the key and what that tells you. For example, we'll have a look today. There's a question that talks about the median viral load. So that's the header that you would use within your own answer, the median viral load. We might have a header that says the mean concentration of product, so we'd have to use that in our answer. Now, when it comes to data, it's really important to be able to look for differences in data and circle the differences. Does a value go up or does it go down compared to another value? So it might be, for example, that we have 2 different concentrations of product at 2 different temperatures. So we're going to highlight the differences, and we might have, for example, standard deviation bars that we can then say that these differences are significantly different, higher or lower compared to the other value. So once we've looked for differences in data, we've circled the differences, we can then start to use our knowledge from the spec to be able to explain the differences. So with the 2 or 3 marker, normally what happens is you'll make some sort of statement as your first mark point. There's more of this or there's less of this compared to A versus B. And the second and third mark points might be then, so therefore the consequence of what I've just said is this. So it's about identifying a difference in the data and then writing about what the consequence of that is. Now the other mark point that you want to look at is AO3. This is an evaluation question. So here you'll be given an experiment and the data and the information of that experiment, and you'll be given either an hypothesis or a conclusion. So the first thing I'd recommend is to be able to write that hypothesis or conclusion yourself in the margin. Now if you write it out yourself, it just gives you that little bit of thinking time to start thinking about, does this data support that or not? So what we're going to do is go back into the data. It might be a table or a graph or a combination of the two. We'll try to pick out some supporting data that supports that hypothesis or conclusion. We might even look at a bit of negative data that contradicts that particular hypothesis. So if we had a hypothesis that said there's an increase in reaction rate with an increase in temperature, the positive data might support that up to the optimum temperature for that particular enzyme, but above the optimum temperature for that enzyme, we know the tertiary shape starts to change for the enzyme and the reaction rate goes down. So that would be the sort of contradicting data against that hypothesis. Now, the third thing that you want to do for an evaluate question is to look for any missing data or any limitations of the experiment. So missing data might be, for example, that the experiment was only done on 1 species of insect, maybe like a bee, species of bee. but the conclusion is referencing all different species of insects. So we don't have the data for all different species of insects. We only have the data for one species of insect, like the bee. Limitations might be there's no statistical test that's been done. So there's no p-values. So we've got no idea of significance or not. So they're just some tips that you might want to read through just before we start going through the answers. Okay, so questions 1 to 6 then. We're going to start with question 1, and it says, describe how a non-competitive inhibitor can reduce the rate of an enzyme-controlled reaction. So if we have a quick look at this, we know non-competitive, that suggests to us that the enzyme has an allosteric site that's able to bind the inhibitor. So the inhibitor is not going to bind at the active site, it's not going to compete for the active site, but it's going to bind away from the active site at the allosteric site. And it says reduce the reaction rate, so that's going to go down. So we know then, if we have a look at the answer, that as the inhibitor attaches away from the active site at the allosteric site, so I would definitely reference the allosteric site, the tertiary shape of the enzyme is going to change, which then changes the shape of the active site. That means it's not now complementary, so it's not complementary to the shape of the substrate. Now, because of this, the consequence that we just mentioned, the consequence is there's less or no enzyme substrate complexes because the substrate cannot fit into the active site. It cannot successfully collide with the active site, which produces the reduction in reaction rates. Now, just bear in mind, okay, AQA these days, they will not accept abbreviations of enzyme substrate complex. So they will not accept ESC. You have to write out enzyme substrate complex. So in this case, the consequence of what we've just said, that the active site is not complementary to the shape of the substrate, the consequence is less or no enzyme substrate complexes. Okay, second part to this question then, it's talking about this molecule called pectin. Pectin is a polysaccharide that we find in cell walls of plant cells, and it helps to bind the cellulose of the cell wall together. Now, scientists investigate the effect of pectin on the hydrolysis of lipids by an enzyme called lipase. So we know here that the lipid is the substrate and the lipase is the enzyme that hydrolyzes the lipid or the triglyceride into fatty acids and glycerol, so we can hydrolyze the ester bonds. Now, if we look at the graph that we're provided with, obviously we've got the x-axis along the bottom and the y-axis across up the left-hand side. Now, if we look at the x-axis, we can see that it says lipid concentration. So this is the same as saying the substrate concentration for lipase. So you should really identify to start with that the x-axis has the substrate concentration, or the lipid concentration in micromoles per decimeter cubed. Now, what they've done here is they've had separate enzyme mixtures, and each enzyme mixture has had a slightly different concentration of substrate. They've had 2, 4, 6, 8, and 10 micromoles per decimeter cubed of lipid. Now, in each of these separate enzyme mixtures, what they've done is, to start with, they've had no pectin, but they've looked at the activity of lipase on the y-axis. So if we took 2 micromoles per decimeter cubed, that would give us around about 0.6 arbitrary units. If we had 4 micromoles per decimeter cubed of substrate, that would give us a value of approximately 0.8 arbitrary units. If we had 6 micromoles per decimeter cubed, that would give us a slightly higher value of lipase activity in that mixture. But then as we get to 8 and 10 micromoles per decimeter cube, what we can see is that the lipase activity starts to plateau or remain constant. So at very high substrate concentrations, we know the enzyme's a limiting factor. So all the active sites of the enzyme are saturated or fully in use, 100% in use, which is why the activity plateaus at the very high substrate concentration. Now, if we had a scenario whereby we had a competitive inhibitor, what you'd expect to see is that as the substrate concentration is increased in a mixture, the lipase or enzyme activity gets nearer to the value with the absence of inhibitor. So this would be a competitive inhibitor with this dotted line that I've drawn on, because at very high substrate concentrations, you would expect the substrate to outcompete the competitive inhibitor for the active site, and therefore you'd have lots of enzyme substrate complexes, and the reaction rate will be much nearer to no inhibitor. So no inhibitor. However, if you look at pectin when this has been added, we can see actually that if I just put some data points here, even at very high substrate concentrations, the lipase activity is kept very low. That indicates there's very few enzyme substrate complexes. Now that's because the tertiary shape of the lipase enzyme has already been altered by the pectin because it is a non-competitive inhibitor. So with a non-competitive inhibitor, you cannot just dilute out the effect of the inhibitor by having a high substrate concentration because the enzymes have altered tertiary shapes. Question 1.2 says, use figure 1, so we have to use the data that we're provided with to explain why the scientists concluded that pectin is a non-competitive inhibitor. And the answer that we'll see next is that even at the very high substrate concentrations, there's a very low lipase activity. It's about 0.1 arbitrary units, and it does not increase nearer to the value with no inhibitor. So just there's the answer then, if you guys just want to quickly go through it. So if you increase the substrate or the lipid concentration, this does not increase the rate of reaction of lipase, or we could say does not increase lipase activity because we are given that on the y-axis. They did accept a high concentration of substrate does not overcome inhibition by a non-competitive inhibitor. You cannot simply dilute out the effect of the inhibitor if it's non-competitive. Or they said a high substrate concentration or lipid concentration does not meet the maximum rate of reaction by lipase. So any of those 4 marks were accepted for that 1 point. Now the second part to this question then, it's talking about pectin stops the action of bile salts. Now we know that pectin is the non-competitive inhibitor of lipase, but now we're being told it also stops the action of bile salts. So the scientists made 2 samples, A and B, and he added lipid and bile salts to A and lipid and bile salts and pectin to B. So in this experiment here, the only difference between the 2 suspensions is the presence or the absence of pectin. So the independent variable for this experiment is plus or minus pectin. Now it's really important to look at the sentence underneath. It says he did not add lipase to either suspension. So we can't talk from this point onwards about the activity of lipase because it doesn't exist in the mixture. It's just not there. So this is about really paying careful attention to what we're given in the text. What we could say at the top, if we just want to annotate this a little bit further, we could just say no lipase. So we're only looking at minus pectin or plus pectin. Now, if you look at suspension A, we can't see any oil droplets. However, if we look at suspension B, we can see oil droplets. So we could actually annotate on here and we could say no oil or lipid droplets. Now we know in suspension A, the bile salts are not being inhibited. So therefore, there is emulsification of the oil or the lipid. Now we know emulsification is where the surface tension of the oil droplets or the lipid droplets is reduced, the surface tension is reduced, so the bigger oil droplets split into thousands of smaller oil droplets with collectively a larger surface area to volume ratio. So this question is only about emulsification, it's not about lipase action. Okay, now it says he observed samples from the suspension using optical microscope. Now an optical microscope is just a light microscope. Now we know a light microscope uses beams of light to shine through a sample. Now because the light has a longer wavelength than, for example, electrons, which have a shorter wavelength for the electron microscope, the light microscope has a low resolution. Okay, So if the large oil droplets have been emulsified into thousands of much smaller droplets, we can't see them in suspension A on the left because of the low resolution of the light microscope. Now, the bile salts are not working in suspension B, and we know they're not working because we can see the oil droplets and there's been no emulsification. Now, I just want to quickly talk about working out the actual size of an object. So if you take your ruler, like we've got on the slide here, and you measure the length of the oil droplet X, you can see hopefully that it's 4 millimeters long. Now we have to convert millimeters into micrometers because we're going to see in a minute that's the units that we're being asked to give. So that would be 4,000 micrometers that we've measured with a ruler. Now this is the image size with a ruler, 4,000 micrometers. That's the image size. Now, if you look at the bottom, we're also given the magnification of 500. So if you want to work out the actual length of the oil droplet, that's A, you can use the IAM triangle. Now, A is the actual size, which is always going to be smaller than the image size. So image size is 4,000. M is 500. So to get A, we're going to go I divided by M. So the next bit says calculate the maximum length of the large lipid droplet X. It says using a ruler with a millimeter interval always does include an uncertainty in the measurement. Use the uncertainty in your measurement to determine the uncertainty of your calculated maximum length. So the first thing we have to do is calculate the maximum length. So let's just write this out again. So image was 4 millimeters. That's 4,000 micrometers. M was times 500. We're going to use the I triangle. So A is I divided by M. We need micrometers. So the golden rule is to convert your units. So it's 4,000 divided by 500, which is 8 micrometers. Then it says calculate the uncertainty in your maximum length. Now it says there's no uncertainty in the magnification. That means when you convert the actual length to the maximum length, you don't have to worry about the magnification having uncertainty. It's just a ruler. If you take a ruler and it's in millimeters, what you can say is that on the left side, you have plus or minus 0.5 millimeter uncertainty in your measurement. And on the right side, you have plus or minus 0.5 millimeter. So with a ruler then, when you measure a line or a scale bar or any distance with a ruler, the uncertainty is plus or minus 1 millimeter. Now we measured the oil droplet at 4 millimeters. So the uncertainty is plus or minus 1 millimeter divided by 4 millimeters. Now that gives us plus or minus 25% of uncertainty. Now we said earlier that our actual length was 8 micrometers. So we've got a plus or minus 25% uncertainty. So it's actually asking us for micrometers for the uncertainty. So 25% of 8 is plus or minus 2 micrometers. There you go. So the maximum length was 8 micrometers and the uncertainty was 25% of that, which is plus or minus 2 micrometers. Now they did accept if you'd measured the oil droplets slightly bigger than 4 millimeters, like 4.5 millimeters, they would have accepted 10 micrometers for the maximum length. So the take-home message is the uncertainty is plus or minus 1 millimeter for the ruler. Divide that by your measurement is 25%, so it's plus or minus 25%. 25% of the actual size is 25% of 8, which is plus or minus 2 micrometers. Okay, the next question then. So it says there's no large lipid droplets visible with the optical microscope in sample A. So we can just put there a light microscope, and we know straight away that's got a low resolution, and we know why, because of the longer wavelength of light. So no large lipid droplets. So it's a 2 marker, so we need 2 answers here. It says explain why. Well, we know the bile salts are not being inhibited because there's no pectin in A. They emulsify the large lipid droplets into lots of smaller lipid droplets collectively with a bigger surface area to volume ratio. And we know the tiny lipid droplets cannot be seen because the light microscope has a low resolution because of the longer wavelength of light. Okay. Due to or because of the low resolution, due to the longer wavelength of light. Okay, question 2 then. So it says the table shows the cell wall components in plants, algae, fungi, and prokaryotic cells. Now, we know prokaryotic cells straight away are, well, one example are the bacteria. So all bacteria are types of prokaryotic cell. And it says complete the table by putting a tick where the cell wall component is present. So we know plants have a cell wall made of cellulose, so they've got straight chains. So a cellulose molecule is made of a straight chain of beta glucose, and that's each second one is inverted 180 degrees. The cross links are hydrogen bonds, and that makes microfibrils, which gives the cell wall in plants its strength, high tensile strength. Algae, we know, also are producers, and they have chloroplasts just like the plants do, so their cell walls are made from cellulose. Algae tend to be unicellular, but they can be multicellular, but they are eukaryotic. They do have organelles like chloroplasts. They are producers. So that will link with the energy transfer topic that we're doing currently. The fungi, they are definitely not plants and they're not algae. They do have cell walls and they're made of chitin. And the bacteria, or the prokaryotic cells, we should say really, their cell wall is made of murin or peptidoglycan. So you can say murin or peptidoglycan. So you should have had a tick in each of those boxes there to get 3 marks out of 3. Okay, for this question we are given some data on a graph and we're given the labels on the x-axis across the bottom and the y-axis going up the left. We're also given a key that we need to look at very carefully. So if you look at the key, it says the data points that have been plotted on the graph represents the mean relative risk of developing cardiovascular disease. Now the dotted line on the graph as well represents the line of best fit to show plus or minus 2 standard deviations above and below the mean value, or each plotted mean value. And the sample size for each of those data points is 1,000 people. So we know straight away there's a large sample size here, so that's not going to be one of the missing or the limiting factors of this experiment. Now it says above that the relative risk of 1 is no difference in the risk of developing cardiovascular disease between the sample of 1,000 people and the population. The relative risk of less than 1 means that the relative risk of developing CVD, cardiovascular disease, is less likely in the sample versus the population, and a relative risk of more than 1 means it's more likely that in the sample of 1,000 people for that data point there's going to be cardiovascular disease versus the whole population. Now if we look at the actual data points that have been plotted, these crosses that have been carefully put on the graph here, hopefully you can see that above and beneath each of them, where that dotted line is going to be, is the standard deviation. So the standard deviation bars, you can actually draw some of them in just to represent them. The top of the SD bar would be where it hits the pink line, and the bottom of the SD bar again would be where it hits the pink line underneath the mean value. So if we selected 2 data points, for example, 6 grams of fibre per day. So the x-axis is the grams of fibre consumed per day. Put X there, put Y on the left. We can compare that with 10 grams of fibre per day. We can see the SD bars do overlap here, and therefore there's no significant decrease in the relative risk of cardiovascular disease between 6 grams or 10 grams of fibre per day. So that's just looking at the data just to see what's going on. If we look at some of the other plotted points, maybe 37 grams of fibre per day, compare that with 45 grams of fibre per day, we can see again the SD bars overlap, therefore there's no significant decrease in the risk of cardiovascular disease. And again, if we looked at 60 grams of fibre per day being consumed, we can see, yes, the mean relative risk of cardiovascular disease has decreased. There's definitely a correlation between the increased fibre consumption and the decrease in the relative risk of cardiovascular disease, but we can see the SD bars do overlap, therefore there's no significant decrease. Now, if you look at the mass of fibre consumed per day, that's going to be measured for each person within each population investigated, so each of the 1,000 people. So this is one measured variable on the x-axis, and then we've got another measured variable, which is the relative risk of cardiovascular disease. So anytime you've got two measured variables, one on the x and one on the y-axis, the second one on the y-axis, we know we might be looking for a correlation. So we can see here, as the mass of fibre consumed per day increases, there's a correlation with a decreased risk of cardiovascular disease. Now we call this a negative correlation. We've got two measured variables, one on the x-axis and one on the y-axis. We can see as the x-axis variable increases, there's a negative correlation and the measured risk of cardiovascular disease decreases on the y. So we can see there those two mean CVD risks, there we go, overlapping SD bars, no significant difference. Now, if we took, for example, 10 grams of fibre per day, so if we took this one on the bottom up 10, we can see that there's a relative risk of cardiovascular disease and it looks to be about 1.2. The mean value is about 1.2. If we then increased the mass of fibre consumed per day from 10 to 20 grams per day, so we're going to go up by 10 on the x-axis. Hopefully you can see that this is the mean risk. It's actually under 1 this time, so it's definitely less than 1.2. But the SD bars are so small for this mean value at 20 grams of fibre per day that we can't actually see them on the data because the SD bars are so small. So there'll be no overlap in the SD bars between the relative risk of cardiovascular disease at 10 grams of fibre per day versus that at 20 grams of fibre per day. So adding 10 grams of fibre to your diet, if it starts at 10 grams of fibre per day, would significantly reduce your risk of cardiovascular disease. Now, if we actually took some other values and we said 40 grams of fibre per day is what we started with, that would be our data point. Our SD bars would be here. If we increase that by 10 grams to 50 grams per day, that's the relative risk there. So the relative risk is actually about 0.7. But the SD bars, if we drew them in with a pencil like that, we can see they do overlap. So if you started at 40 grams of fibre per day and increased by 10 to get to 50 grams per day, you would have a reduced risk of cardiovascular disease, but it would not be a significant decrease. So we can start to use this data to answer some of the questions. So it says, a student concluded from figure 3 that eating an extra 10 grams of fibre per day would significantly lower his risk of cardiovascular disease. So this is the AO3 hypothesis question. So they're saying an increase in 10 grams of fibre per day significantly lowers CVD risk. So we're just going to spell out or write out the conclusion again. Now, the first thing hopefully you can recognise from this is that it says eating an extra 10 grams of fibre per day significantly lowers cardiovascular risk. But what we don't know is the starting value. So we don't know the starting grams per day of that person. It could be 10 grams per day to start with and you increase to 20, or it could be 40 grams per day and we increase to 50. That is missing data that we're not provided with. So let's evaluate this conclusion. We're going to start with positive data, and then we're going to look for any negative contradicting data, and then we'll start looking at the missing data or limitations of the experiment. So the first mark point then at the top, it says there's a negative correlation that we mentioned, and you have to use the word correlation because it's two measured variables, between more fibre eaten per day and a reduced risk of cardiovascular disease. However, mark point number 2, we do not know the original or current fibre intake for that person. So we don't know if it's 10, 20, 30 or 40 grams or more per day. It doesn't tell us that. That's missing data. So we can put missing. Now, the third mark point was for using the standard deviation bars above and below each mean value, as indicated by the dotted line on the graph. And it says there's this idea of significance or a significant reduction in risk is linked to no overlap between the two standard deviation bars above and below each mean, or there could be overlap between SD bars and therefore there would be no significant difference in means. So any reference to the use of SD bars overlapping no significant decrease or not overlapping there is a significant difference or decrease will get you mark point 3. Now for mark point 4 then, you can either have a current intake of between 5 and 30 grams per day. So you could say, for example, 10 grams per day to start with. If you increase that by 10, because that's what it says in the conclusion, to 20 grams per day. Okay, there is no overlap in SD bars, therefore there is a significant drop in risk. Or you could have said between 30 and 50 grams per day if you increase by another 10. So imagine you start at 40 grams per day and you increase that to 50 grams per day. Okay, there is a reduction in risk of cardiovascular disease, but it is definitely not significant because the SD bars overlap. So it depends on the starting intake of fibre per day. Now we've said there's a negative correlation, so we've said negative correlation for mark point number 5. However, we know correlation does not mean causation. Now you can't just say that in the exams these days. What you have to say is it's not necessarily an increase in fibre or mass of fibre per day that reduces the risk of cardiovascular disease. There could be other named factors that may also contribute to a lower risk of cardiovascular disease. So it might be, for example, that if someone has more fibre per day in their diet, they also do more exercise, or they do not smoke, or they drink less alcohol, or some other factor that could also impact the development of cardiovascular disease. So that would be a confounding variable where there's other factors that can contribute to the development of cardiovascular disease. So it might be that if someone has less fibre per day, they also smoke more or have more alcohol or do less exercise. That could also correspond to the increased risk. Now, also we could have mark point number 6. So there's little evidence or little data for the higher mass of fibre per day. If you look at the graph, you can see lots of data points for around 10 to 20 to 30 grams of fibre per day. There's actually many fewer data points for 40, 50, 60 grams of fibre per day. So there's less data available for the higher mass of fibre eaten per day. Less data points. Now, the larger standard deviations for very low fibre eaten per day or very high mass of fibre eaten per day means that there's a greater spread of data around that mean value. So there's greater variation in the data from that sample of 1,000 people. So greater, could say greater variation in some of the data points, especially at the higher fibre, or greater spread of values away from the mean because of the big standard deviation bars. And the third one, even though we did have plus or minus 2 standard deviations above and below the mean, we can actually then incorporate or bring in a p-value of less than 0.05 or greater than 0.05. We can actually say that. However, we don't have a measure of the actual p-value. So to do that, we'd have to do a stats test such as the t-test. Now a t-test is used to compare two different mean values to see if they're significantly different, and we'll get a p-value, and it might be p equals 0.0001, which means there's a very high significant difference in the two means. So there's no t-test or no stats test to give us specific p-values. Okay, question 2.3 then. The scientists estimated the mean mass of fibre eaten per day using a food frequency questionnaire. So the FFQ basically is a questionnaire that people answer on a piece of paper, and it asks people how often they have eaten many types of food over the past year. Now, if you compare that to the alternative method, it says there's a nurse that asks one-to-one face-to-face questions to the person, a detailed set of questions about what they have eaten in the last 24 hours. So the FFQ is a questionnaire on a piece of paper asking people to try to remember what they ate over a year. And the alternative method is a nurse asking very detailed questions over the last 24 hours. Then it says, suggest one advantage of the FFQ and one disadvantage of the FFQ compared with the alternative. So if you highlight both of these sentences in different colours, you know that you're focusing on the first one at the top. So the advantage of having a questionnaire whereby people put in what they've eaten over 1 year is that if you have data only for 24 hours, that person might have only eaten certain food types in that 24 hours, and it's not representative of what they would eat over the course of an entire year. So one advantage of the FFQ is that there's more representative data because it's over 365 days as opposed to a single day. So it gives a much more representative indication of what they've eaten and what their diet is like from day to day as opposed to on a single day. Now, one of the disadvantages of the FFQ then could be that it's not as accurate. So people have to remember what they ate 2, 3, 4, 5, 6 months ago, 12 months ago, and that's going to be less accurate data compared to the very accurate data obtained by the nurse asking very detailed questions of what they've eaten very recently. Let's just have a quick look at the answers that were acceptable, then you can pause the video obviously to go through it. So the advantage of the FFQ was that it's over a very longer period of time, therefore the data is much more representative of what they've actually eaten over the year. The diet can change from day to day, it's not the same every day. A person is more likely to be honest on a questionnaire because they might not want to say certain things to a nurse face to face. Maybe they've had, things they shouldn't have eaten. Maybe they had too many cakes, etc. So they're much more likely to be honest on a questionnaire than a one-to-one interview with a nurse. You could say it's more cost effective because obviously the FFQ does not use any nurses in this, so there's less personnel needed to run the experiment. However, the disadvantage then, so let's separate the two. It relies on long-term memory, so therefore the data that's acquired may be less accurate. So the first mark point at the top was about being more representative, but the second mark point is about being less accurate. So they're less likely to recall what they've had over a year compared to a 24-hour diet. Now, they did accept that the person may be more honest when being interviewed by a the person may be more honest when being interviewed, but they would only accept honesty once, as is given up at the top here. So question 3 says a group of students investigated biodiversity in different areas of farmland. So they've had 3 different areas. So let's circle this, 3 different areas: the centre of a field, the edges of a field, and the hedge between the different fields. So then they've plotted some data in figure 4. So we've got the y-axis is the index of diversity, and the x-axis is categorical. So it's the center of the field, the edge of the field, or the hedge in between different fields. Now we can see index of diversity on the y-axis. The hedges between fields have a greater index of diversity of 0.96. The edge of the field has a lower diversity, and the center of the field only has has the lowest of the index of diversity values. Now, just to remind ourselves, okay, the equation for this index of diversity is as has been drawn on the slide. So big N is the total number of individuals for all the different species, and little n is the number of individuals for each separate species in that area. So we're going to see later it's going to ask us about either putting more hedges in the farmland or removing the hedges, so we're going to have to use this data in a minute. Now, one thing to note is that it doesn't actually tell us what's been measured, what populations. We don't know if it's plants or is it animals or is it both. Now, we know that there's 3 methods that we can use for investigating populations. If it's plants in a defined area, we can use random sampling with random coordinates and quadrats. If it's plant species along a transect from A to B, where we look at some environmental gradient, it's going to be a line transect with quadrats at equal intervals. Or if it's animals that move around, we have to use the mark, release and recapture method. So calculating an estimate of population size, we'll use 3 different techniques. Okay, so what data would the students need to collect to calculate their index of diversity? Do not include apparatus such as random sampling and quadrats used for species sampling. So all we need for index of diversity is the number of different species present and the numbers of individuals of each of the different species. So we need the number of different species present, maybe there's 10 different species, and the numbers of individuals of each of the 10 different species. Now, the numbers of individuals of a given species in a given area at a given time is the population. So the population refers to one specific species. So we might have 10 populations of 10 different species, 1 population per species. So you could say all the individuals of all the different species present in a given area at a given time. Now, give 2 ways the students would have ensured their index of diversity was representative of each habitat. So there's 2 main ideas with a representative sample. One is that the sample size is very large. So if it was random sampling, you'd have 20 quadrats minimum, or you'd have enough quadrat samples where the running mean was stable, but you do need a large sample size. And the second one is that the samples have to be random, and that reduces bias in the data set. So random samples using a random number generator would create data with no bias, so that's really important. And large sample size means the sample is a representative sample with a reliable mean, or you could say stable running mean, and you have to say stable on that one. Modern farming techniques have led to larger fields and the removal of hedges between fields. Use figure 4 to suggest why biodiversity decreases when farmers use larger fields. Right? So the stem of the question says the removal of hedges. So biodiversity goes down when farmers use bigger fields because they've removed, up here, they've removed the hedges. But we have to use figure 4. So if we look back at figure 4, hopefully you can see that if you remove hedges, hedges have the biggest index of diversity. So we're going to have less hedges with the bigger index of diversity. And if you take away the hedges, the center of the field, the area of the field, particularly the center of it, is going to increase. Now, the center of the field has the lowest index of diversity at 0.85. So if you create a bigger center of the field by removing the hedges, it's going to have a lower index of diversity. So you're taking away or removing the hedges, so there's less hedges. So you're taking away the habitats which have a greater index of diversity, and you're creating more habitats like the center of the field, which has a lower index of diversity. So you've got to use the labels here. So when you when you take away more hedges, they're going to go down, and it's going to make the center of the field bigger. So you could have said more center of the field, less hedges, or less edges of the field, or fewer species present, were all acceptable. Now you can't just say removing more hedges or more hedges are removed. That is in the stem of the question, and you had to use the figure to help you with that answer. Okay, farmers are now being encouraged to replant hedges on their land. That means put more hedges back, replant them. Suggest and explain one advantage and one disadvantage of a farmer of replanting hedges. So suggest is make some sort of statement, and explain is say why, to explain the importance of what you've just said. Now, to a farmer, so a lot of students miss the idea of the advantage or the disadvantage being to the farmer. So you might want to talk about the yield of crop that he can harvest. It might be about the profit that he gains or the lack of profit, but it has to be in relation to the farmer. So you can't just say there's a higher index of diversity, there's a greater number of species present, because it's not in relation to the farmer. So let's have a look at the advantages then. The greater biodiversity means, so therefore, the consequence of that is an increase in the population of predators that might feed off the pests that would feed off the crop. So it allows the crop to grow and not being eaten by pests. We could say increase in biodiversity because they've planted more hedges, increases the predator population of pests. So therefore, there's more yield or more income or more profit margin for the farmer because they have to use less pesticides because the pests are being eaten. Or we could say an increase in biodiversity because we planted more hedges increases the number of pollinator individuals, or maybe there's a greater number of pollinator species, or maybe there's a greater population of a specific species of pollinator. So therefore, the consequence of that to the farmer is a greater yield and therefore a greater income. Or, last one accepted, more tourists, more diversification on the farm, more subsidies from the government to fund more farming. So therefore, the consequence of that is, again, more income to the farmer. Okay, next one then. Describe the structure of the HIV virus, human immunodeficiency virus. Now this is recall A01. So if you look at the picture that you guys have got in the immunology topic, HIV is a retrovirus. So the nucleic acid is RNA, or ribonucleic acid. There's actually two molecules of single-stranded RNA. The RNA is kept inside a protein shell called a capsid. So you have to reference that the capsid is made of protein. Now inside the capsid, as well as the RNA, are certain enzymes, one of which is called reverse transcriptase. The other type of enzyme is actually called integrase, and we'll see what that does in a minute. Outside the protein capsid is a lipid envelope or a lipid membrane or a viral membrane or a viral envelope. Any of those terms are accepted. Now that originates from the cell membrane of the host cell, but we can't call it a cell membrane here because obviously a virus is non-living. So we call it a lipid membrane or a viral membrane or a viral envelope. Now within there, we actually get glycoproteins. Now the glycoproteins are actually HIV proteins. And these HIV proteins are actually part of the capsid. They would extend out from the capsid. They are called attachment proteins, and they actually attach to the receptors on the T helper cell. Now the receptors in the cell membrane of the T helper cell are the receptors to which the glycoproteins, the attachment proteins, bind to allow HIV to go into the host cell. So the host cell in the human body for HIV is the T helper cell. So we can see there then that RNA was given 1 mark. There's actually 2 molecules of RNA inside the capsid. There's an enzyme called reverse transcriptase. There's the protein shell called the capsid. There's a lipid or viral envelope that surrounds the capsid that originated from the host cell membrane. And there are glycoprotein attachment proteins that will bind to the host cell membrane, to receptors in the host cell membrane. Now, if we look at the enzyme reverse transcriptase, that's number 2. When HIV injects its RNA into the host cell cytoplasm, the reverse transcriptase uses DNA nucleotides and links them together into a chain of single-stranded DNA. So this is the opposite of transcription, or the reverse of transcription, because transcription is normally the production of messenger RNA against the template of DNA. Well, here, single-stranded DNA has been made against the template of single-stranded RNA. Now, what else happens with the reverse transcriptase is that the single strand of DNA separates, and against that, a second strand of DNA is made that's complementary to the original first strand. So this is now called, we can change that now, double-stranded DNA, and this is HIV DNA. Now, the other enzyme that's present is called integrase, and that inserts the HIV DNA into a host chromosome inside the nucleus. Now, this is why some people with HIV infection do not develop symptoms of AIDS for quite a long time after being infected, because the HIV DNA inserts into a host chromosome, and if that chromosome or that part of the chromosome or the DNA is not being transcribed, then the HIV RNA is not being made, and therefore the HIV proteins are not being made, so they can't make HIV particles. So the HIV DNA, which is made using reverse transcriptase, inserts via integrase into a host chromosome. And once that DNA is transcribed using the normal mechanisms of transcription inside the nucleus, HIV RNA is then made again, which will allow HIV particles, virus particles, to be made. Okay, next question then. So it says some people infected with HIV do not develop AIDS. Now, this doesn't mean that people called HIV controllers don't have HIV. Yes, they do. They're still infected with the HIV virus. It's just that they haven't developed to the later stages of HIV infection called acquired immunodeficiency syndrome. So they don't have the characteristic symptoms of AIDS. So they're called HIV controllers. So scientists measured the number of HIV particles called the viral load in the blood of these patients, and they measured the number of T helper cells in the blood of both groups of patients. Now we know when we get a table, we must use the headers. So it says for the first measurement, they took the median viral load in 1 centimetre cubed of blood. And the second measurement was the median number of CD4 cells, which are T helper cells. Now you should know that HIV infects T helper cells as the host cell. We'll put host cell. So over time, the T helper cells can get infected with HIV particles, and it leads to the destruction of the T helper cells. Now, if we look at the data then, we can see HIV controllers, they have 212 virus particles per centimetre cubed of blood. Now, if you look at, because we're looking for differences in data, the median viral load in HIV positive people with AIDS, that's actually much, much higher. It's 66,274. So we're comparing 212 with 66,274. So we can see HIV controllers have a much lower median viral load compared to people with AIDS. Now, if you look at the median number of T helper cells or CD4 cells per millimeter cubed of blood, it's the opposite. So 693 on the right with the blue arrow is actually a lot higher than 248. So people with HIV, called HIV controllers, they have a lower median viral load in their blood versus people with AIDS, but they have a higher T helper cell count in their blood compared to people with AIDS. Now, if we go back to the left-hand column, mean viral load, hopefully you can see that within this sample of patients, the minimum value is less than 50 and the highest value of those patients was 609. Right? So this is the range. The range is the minimum to the maximum value in that data set. Now, if you have a look at the HIV controllers, the highest number of virus particles in the blood for one of these patients was 609. That is actually a lot lower than the lowest value in the sample of people with AIDS. So the range in the sample of people with AIDS was 30,206 up to 306,163. So the lowest value of any of the patients with AIDS was actually higher than the highest value in the HIV controllers, and that's for the viral load. Now, the mean number of T helper cells or CD4 cells, we can see hopefully now that in the range, the minimum value of 529, so one patient had 529 T helper cells per millimeter cubed of blood. That is higher than the highest value of the patients with AIDS. So even in the patients called HIV controllers, their lowest value was 529, which is higher than the highest value of the patients with AIDS. So we're going to use this data to help us in answering the questions. So it says a test sample of 500 millimeter cubed of blood is taken from an HIV controller to determine the viral load. Tick one box that shows the number of virus particles that will be present in a test sample of blood taken from an HIV controller with the median viral load. So if you go back, we know that it tells us the median viral load was 212 in 1 centimeter cubed. That's just using the header. Now 1 centimeter cubed is a volume. So it's 1 centimeter by 1 centimeter by 1 centimeter. Now that's equal to 10 millimeters by 10 millimeters by 10 millimeters. So 1 centimeter cubed is 10 by 10 by 10. That's 1,000 millimeter cubed. Okay, but it says in 500 millimeter cubed. Okay, so it's going to be half of 212, which is 106. So we're not looking at 1 centimeter cubed, we're looking at 0.5 centimeter cubed, which is 500 millimeter cubed here. Okay. So the answer was 106. Right, use the data in table 3 and your knowledge of the immune response to suggest why HIV controllers do not develop symptoms of AIDS. Okay. So again, we can see from the data that the median viral load is 212. The highest value is 609. Okay, so this is the viral load at the bottom here. So the number of free particles of virus in the blood. And the lowest value of the patients with AIDS was 30,206, which is much, much higher than the highest value from the HIV controllers. So every patient who was a HIV controller, right, had lower numbers of virus in their blood compared to every patient with AIDS. Now, if we look at the other set of data, so this is the T helper cells. Okay, the biggest value is 887. The smallest value is 529. So in these patients, HIV controllers, the lowest number of T helper cells was 529. Okay, and the highest number of T helper cells in the AIDS patients was 365, which is lower than the biggest number of HIV controllers. So what we can say is HIV controllers, all of them, have a smaller viral load compared to the people with AIDS, and they have more T helper cells compared to all the patients with AIDS symptoms. So let's have a look at the marks then. So mark point number 1, all the HIV controllers have more T helper cells compared to the AIDS patients. They also have lower viral loads, all of them, compared to the AIDS patients, and therefore there's less virus particles to infect or destroy the T helper cells. So yes, they have more T helper cells, which is brilliant because that means the cell-mediated response can take place. They have a lower viral load, they have less virus in their blood, therefore there's less virus to infect or destroy the T helper cells, of which there's more anyway. Now, because there's more T helper cells, you can then talk about what they do. So if a person is infected with a pathogen, we know the B cells, they act as antigen presenting cells, and they present antigen to the T helper cell. The T helper cell secretes cytokines, and they allow the B cells to undergo clonal selection. So activation of B cells via clonal selection, that's going to form lots of clones. Of the clones of the B cell clones, half of them differentiate into plasma cells, whose job is to secrete antibodies into the blood. So now we're getting the humoral response because we've got working T helper cells, or enough of them, in the HIV controllers. And therefore, if the person who is an HIV controller can make antibodies to destroy pathogen, there's less opportunist infections in HIV controllers, so they do not develop AIDS as quickly. Now, they also did accept that if there's more T cells, there might be more cytotoxic T cells, and we know the function of cytotoxic T cells in the HIV controllers is to kill directly any virus-infected cells. So any mutated or cancerous cells or virus-infected cells can be killed by cytotoxic T cells. So this question was really about understanding that there's less virus to infect the T helper cells. There's higher numbers of T helper cells present. Therefore, there's more of the cell-mediated response to activate B cells via cytokines. B cells divide by clonal selection, by mitosis. Some of those form plasma cells, which are specialized and differentiated, and their job is to make and secrete lots of that protein, antibodies, into the blood, and therefore they can bind to or agglutinate pathogens, and therefore lead into this destruction of pathogens. Okay, next question, in question 5, it says a student used the apparatus shown and the digital balance to determine the rate of water movement in a celery stalk in grams per hour per group of xylem vessels. Okay, so let's have a quick look at this. So grams per hour per group of xylem vessels. Okay, so grams is one measurement we need to make per hour, so time is another measurement, and the number of groups of xylem vessels is the third measurement we need. So we need 3 measurements to determine the rate of water movement. So you've got to pay careful attention to what the information is giving you. Now, We can measure time, obviously, with a stop clock. That's okay. Grams then, well, we know that the leaves lose water vapor through transpiration. So water or water vapor transpires out of the stomatal pores of the leaves down a water potential gradient. So down a water potential gradient to the outside. And we're going to measure basically the loss of mass over time. Now, we're not going to measure the mass of the celery because we don't need to. All we need to do is use this digital balance, and we need to measure the mass of the whole thing initially. So we'll put initial mass, and then we need to measure the final mass after, say, 20 minutes, and then we can get the change in mass. So to work out the loss of water vapor as mass, we just take the whole thing, measure the initial mass minus the final mass, and we're going to get the mass of water vapor that's been transpired out. So the student measured the time taken for water movement, so they've taken care of that. Two of the measurements he made to calculate the rate of water movement. So the initial and the final mass of the beaker and all the contents to work out the mass of water that's been transpired. And the number of groups of xylem vessels, because it says per group of xylem vessels. So if you've got 10 groups of xylem vessels, you could then divide by 10 to get per group of xylem vessels. So that was looking at the information really carefully to get this second point. Okay. Initial and final mass as well at the start. What they didn't accept was, it says reject here, look on the right, the mass of the celery. Okay, we're not measuring the mass of the celery because that's in the beaker of water in the beaker. So to measure the mass of celery, you have to kind of take the celery out and shake it and get rid of the water. You can't do that. So it said reject this point here, the mass of celery. You take the initial mass of the whole device and the beaker and the water and the celery all together, and then the final mass. And that will give you the mass of water that's evaporated through transpiration. Okay, give the reason for adding a layer of oil in the water. So this was really well done. It prevents the evaporation of water, so it prevents water loss from the water in the beaker. All we want to do is measure the water loss through transpiration through the stomata of the celery. So it allows us to measure water loss as mass through transpiration. Okay, a different student used coloured water to investigate the movement of water in the leaf stalks of celery. So she cut equal lengths of stalk from each plant. She put the cut end of the stalk into coloured water. She left them, so we can actually do a little sketch here. We can have coloured water in the beaker. You can have your celery, and the coloured water is going to move up the xylem vessels like this, goes up via the cohesion tension theory. She's then cut 1 millimeter thin slices, and she's looked for the colour, and then she's stopped when the colour disappears or is not present, we should say. Okay. So use a sharp scalpel to cut slices from the stalks at 1 millimeter intervals until she reaches a slice with no coloured water. Explain why coloured water moved up the stalks. Okay, you have to take your time with this one. So we know water evaporates from the air chambers of the leaf and it moves out via transpiration down a water potential gradient. So there's water lost through transpiration from the leaves. Now that's not osmosis because the water molecules, water vapour, diffuses out of the open stomatal pore. However, that water has to be replaced, and it's replaced by water moving out of the xylem vessels down a water potential gradient. So you're going to reference a water potential gradient. Water moves out of the xylem via osmosis to replace the water lost through evaporation. Now, the movement of water out of the xylem down a water potential gradient creates a tension. and that helps to pull the water up. It's called transpirational pull. So the osmosis of water molecules out of the xylem vessels down a water potential gradient creates a tension. Now, a lot of you did write cohesion. Water molecules hydrogen bond to each other. However, you need to reference cohesion in relation to helps to maintain a continuous column of water. Okay, so that last mark point was for saying, yes, there's a tension on the water as water is pulled upwards due to the loss of water via osmosis out of the xylem. Water molecules stick together through hydrogen bonds. They're weak individually, but they're strong collectively, and that provides cohesion or cohesion forces, which maintains a continuous column of water up the xylem, going up the xylem. Now, some of you did mention about the negative pressure. So if it says the rate of water movement up the xylem further up is greater than the rate of water movement up the xylem a bit further down, that's okay, that's correct. And that also creates a negative pressure, which contributes to the upward movement of water through the xylem vessels. So water and minerals only move up the xylem through cohesion tension theory. Okay, the student used a sharp scalpel to cut the celery. Describe how she could ensure she handled the scalpel safely during this procedure. So the procedure is the cutting of the celery. So there's two main points for this then, that she used the sharp scalpel by cutting away from her body. So she's not cutting towards any fingers, for example, always cutting away. And she cuts downwards against a hard, non-slip or flat surface, such as a cutting tile. Now, you guys will do that when you do the heart dissection, and you'll learn how to dissect correctly if, obviously, we're allowed to do that at some point this year. Okay, next question then. So the student measured the distance the coloured water had travelled in 8 celery stalks. Her results were shown in table 1. So if we have a look at table 1, there's actually only 8 values given here. So that's a very small sample size. We need a large sample size to have representative data and have a nice reliable mean value. And it says the student had to choose whether to summarise her measurements by calculating the mean, the median or the mode. Circle the most appropriate measure for this set of measurements. Well, if you look at the data, A, there's a small sample size, and B, most of the values seem to be around 30 or 40 millimetres, but we get 2 outliers or anomalies, 70 and 80. So the temptation is to go for the mean value as the measure of the central value. However, if we think about it and use the data, you know, if you do a mean value with 8 values, 2 of which are very high, that might skew the mean away from the actual true mean value. So if we use the median, what we can do is we can arrange the numbers in sequence in terms of how small or big they are. And the median is basically the middle number. So the benefit of using the median is that it gets rid of the outliers and they don't affect the central value of the data set. Now the mode is the number of times a value repeats, or it's the number of the highest value. So for example, 35, we can see there's 2 35s in the data set and every other number occurs only once. However, that does not take into account the anomalies. So the median would be the one that we'd select. Okay, A, because there's outliers of 80 and 70, which you can see here, and because of the small sample size of only 8. Now, if there's an even number of values, so here there's 8, to get the middle value, what you do is you add together the 2 middle values when you put them in sequence and divide by 2, and that'll get you to 41. Okay, so we could put these in sequence, so you guys can just do this yourself. You can go 30, what's the next one, 35, 35, 40, then 42, then 44, then 70 and 80. Okay, so you go 1, 2, 3, 4, 5, 6, 7, 8. So you take the 2 middle values, add them together, and then divide by 2. I'll get you 41. Right, if you did select mean and calculate the mean as 47, you got a mark, or if you selected the mode and you did the mode as 35, you also got 1 mark, but that was the maximum. Okay, question 6 and last question today. So the scientists investigate the cell cycle in heart cells taken from mice 6 days before their birth and then at 4, 14 and 21 days after birth. So this question is about heart cells and you have to use the context of the question. Now we're going to annotate this table because we need to. It says mice 6 days before their birth. Now age 0 is the day of birth. Now there's no data for age 0. There's only data for days 4, 14 and 21. Now we know these must be after birth. So we can highlight these and annotate the data. If we look at the percentage of heart cells undergoing mitosis, we can see it starts at 13.9 and there's a downward trend. So actually there's less mitosis going on as a percentage as the days progress from before birth, minus 6 days before birth, to 21 days after birth. Now what you can't say here is that the percentage of heart cells decreases in mitosis after birth. There's no data for day 0. If day 0 was given as 13.9, which is the same as minus 6, then we could say after birth there is a decrease in the percentage of heart cells undergoing mitosis, but we can't say that. All we can say here that there is a decrease in the percentage of heart cells undergoing mitosis from minus 6 days before birth up to 21 days after birth. Now, if you look at the data on the right-hand side for the replication of DNA and the percentage of cells in S phase, we can see it's actually a lot lower compared to the value next to it. So all the values on the right are lower than the values on the left. So a snapshot at minus 6 days would give us 13.9% of cells in mitosis, which is PMAT, prophase, metaphase, anaphase, telophase. That's a small part of the cell cycle. And the percentage of heart cells undergoing DNA replication is less at 8.5. That tells us that DNA replication takes place before mitosis does in the cell cycle. So S phase occurs before mitosis. Okay. So if you look at minus 6 days, we can see only 8.5% of cells are undergoing DNA replication in S phase. Now, if you look at day 4 after birth, we can see, yes, actually, there's a smaller percentage of cells in mitosis, which is very similar, well, it's the same here, to the number of cells as a percentage that were undergoing DNA replication. So the growth of the heart is slowing over time from minus 6 days before birth up to 21 days after birth. So we can see at day 4, 8.5% of cells are now undergoing mitosis. 2.6% of cells are undergoing DNA replication. That means a little bit later on, there's a lower percentage of cells undergoing mitosis. Okay, it's similar to 2.6, 1.6. 0.2% of cells are undergoing DNA replication at day 14. Therefore, at day 21, we know there's going to be a low percentage of cells undergoing mitosis at that snapshot because a little bit of time before that, there was a low percentage of cells undergoing DNA replication. And at day 21, this is important, we can see the number of cells undergoing DNA replication is 0. So I would always highlight the 0. No cells undergoing DNA replication at day 21. That indicates that the development of the heart has finished, has completed, it's fully developed at that stage in these mice. Because there's no more cells undergoing DNA replication, the heart is fully formed at day 21. So we've got to use the values that were given. Okay, so there's a trend of the slowing growth of the heart from before birth at minus 6 days up to 21 days in the investigation. We could say the DNA replication happens before mitosis. Really important to indicate that, mark point number 2. Or you could say the heart is slowing down in terms of its growth until it gets fully developed at day 21. Or we could say at day 21, the heart cells have lost the ability to divide because there's no more, this one in, no more DNA replication. So again, you've got to use the heart cells from the mice. -6 days is before birth. 21 days development finishes because there's no more DNA replication and the heart is fully developed. Okay, the scientists determined the percentage of heart cells undergoing DNA replication by using a chemical called BRDU. These cells use the BRDU molecule instead of any nucleotides that contain thymine as their nitrogenous base. Describe how BRDU will be incorporated into a new molecule of DNA using semi-conservative replication. So this is AO2 application. Basically, you've just got to list the activity of semi-conservative replication of DNA, the processes that take place, but you're going to put in the BRDU, okay, during that answer. So we're going to say DNA helicase unwinds the DNA double helix. DNA helicase breaks the weak hydrogen bonds between complementary base pairs. So can you say between complementary base pairs? So that's here, between complementary base pairs. Weak, I would say weak hydrogen bonds. Okay. BRDU mimics a nucleotide with thymine, and it's complementary to the adenine base on the template strand of DNA. Or you could say BRDU forms weak hydrogen bonds with adenine on the template strand of DNA. We know nucleotides with their nitrogenous bases align next to the exposed bases on the template strand. BRDU mimics or actually replaces the nucleotide with thymine, but you have to say it's complementary to adenine and you have to spell it out. You can't just put capital A. Right, the second enzyme, DNA polymerase, joins or links together, we could say nucleotides into the new chain, polynucleotide chain of DNA. So you had to reference it links nucleotides. The DNA polymerase enzyme has nothing to do with complementary base pairs. So the complementary base pairing by hydrogen bonds just takes place with no enzyme. Now the DNA polymerase has an active site that is complementary to the phosphate group of one nucleotide and the deoxyribose sugar of the starting nucleotide, and it will form a phosphodiester bond via a condensation reaction. So you could even put that in, a condensation reaction. So the phosphodiester bonds are the ones that link nucleotides together into the sugar phosphate backbone. Now the mark scheme did say reject hydrolyzes hydrogen bonds. That's a big reject. Okay, the DNA helicase does not hydrolyze hydrogen bonds. It unwinds the DNA double helix and it breaks the weak hydrogen bonds. Mark point number 1 and number 2. Another big reject was that DNA polymerase forms hydrogen bonds between complementary bases. It does not. It links nucleotides into a strand of DNA by forming phosphodiester bonds. Okay, so last question, 6.3, is about cells with BRDU incorporated into their DNA. Now we're going to detect this DNA with BRDU in it by using an anti-BRDU antibody with an enzyme attached to it. So as long as we understand the theory of the ELISA test, we can tweak this a little bit to meet the AO2 application question. So you can write out about the ELISA test, but obviously tweak it a little bit to make sure we include this specific example. So the first thing I would do is to extract the DNA from these cells with BRDU in the DNA. And I would attach the DNA with the BRDU to the inside of a well. So you might have a well like this on a plate and you attach the DNA to the inside of the well like that. This will have BRDU in it. Okay, like that on the inside of the well. You then add the anti-BRDU antibody. So the ends of the antibody, the variable domains, have a tertiary shape. that is complementary to the shape of the BRDU molecule, which is in the DNA. Once we've added the anti-BRDU antibody that binds to the BRDU in the DNA, so the BRDU is basically acting as an antigen here, you have to wash the well with water to remove excess unbound BRDU antibody. Once we've washed off the excess or unattached anti-BRDU antibodies, that's going to prevent us getting any sort of false positive results. We then add the substrate. The substrate is complementary to the shape of the active site of the enzyme. It's going to form an enzyme substrate complex, and it's going to release a coloured product. So the substrate is complementary in shape to the shape of the active site of the enzyme, and it'll be converted into a coloured product. So this will only occur if we get a positive test, and if we've got BRDU in the DNA that we extracted and put on the inside of the well, to which we added the antibody complementary to the shape of the BRDU with the enzyme on the end that can then bind and break down the substrate into a product, which is a coloured product. And that's 3 mark points. So binding and adding of the anti-BRDU antibody to the DNA with BRDU, number 1. Washing off any excess unbound antibody with enzyme, number 2. And adding a specific substrate which will be converted to a coloured product, number 3. Okay, that's part 1 done. So question 7 and 11 we'll go through in part 2. So if you guys could at some point sit down and go through that, that'd be great.